Welcome to the Human Health Exposure Analysis Resource webinar series, webinar number 21, and the final webinar of the series. I'd like to thank everybody attending it. Today's speaker is Dr. Elena Colensino. She's a full professor at Icahn School of Medicine at Mount Sinai. Before the webinar, I'd like to show where the Q&A boxes are. Can we go to the next slide, please? Yeah, so if you have any questions, there is this box, a button here, Q&A. So if you can click on that one and type your questions. So at the end of the webinar, we can take those questions. Okay. Yeah. So it's my great pleasure to introduce today's speaker, Dr. Elena Colensino, a full professor at environmental medicine at Icahn School of Medicine at Mount Sinai, New York. She's a molecular epidemiologist and biostatistical expert. She's PI for several grants on children's health and applying these advanced biostatistical methods for understanding the effect of chemical exposure on children's health. And it's really an honor to have, and thanks for accepting this invitation, Elena. Elena, floor is yours. Please, we're really exciting.
Thank you for having me. It's a pleasure to be here, and let me share my screen so everybody can see. Hope you can see the screen. Let me know otherwise. Okay, so I'm going to talk about immune-related responses in children and the environmental exposures. Oops. So the immune system helps guarding the organism against the exogenous and endogenous stressors that are detrimental for the body, and also supports several processes like neural pruning, triglyceride production and liver regeneration, and blood pressure regulation, interacting with other physiological systems. So it is a major public health priority, understanding why there is inter-individual variability in immune-related responses. The immune system begins to develop, I don't know why it's moving, the immune system starts developing in utero, and as we age, our immune system builds a growing repertoire of antibodies that becomes an integral part of our lifelong immune memory. But as we said, there is variability across individuals in antibody response to vaccination, which is an aspect of the immune system. Environmental exposures have been shown to contribute to reduce antibody responses to vaccination. And here we have an example. We see in the plot on the right, individuals with higher level of a lead exposure that have a lower antibody titers against COVID-19 compared to those that are exposed to lower level of lead. However, the research on how exposures influence immune responses has been under investigated. So the developmental origins of health and disease hypothesis poses that dose and timing of early life exposures can have lasting consequences. So we hypothesize that early life exposure to environmental factors might program immune dysregulation that manifests years later. So the most studied immunotoxic exposure is lead. And lead is often assessed in blood and serum. And blood and serum lead concentrations are indicative of 1 to 6 months of exposure prior to the blood draw. And here in this table, we see that in this national study in children, of 7,000 children, we found that a unit increase in lead level is associated with a decrease in anti-measles and anti-mumps antibody levels of approximately 2 to 3%. And this association is not linear. So we want to identify also not only if lead exposure is detrimental, but also when lead exposure is most detrimental. So to do so, we need repeated collection of samples and lead assessment. But this is very costly and also very invasive for children. So tooth data can provide a new matrix capturing the exposures over time. And we have to describe a little bit the aspects of the tooth development. So teeth start developing during the second trimester of gestation, and the mineralization starts on the cusp tip. Then the enamel and dentin deposition occurs rhythmically, and they form incremental lines similar to growth rings in a tree. At birth, there is an accentuated incremental line, which is called a neonatal line, and this is formed regardless of the type of delivery. And after birth, teeth continue to grow and to show this growth line. So through this mineralization process, the exposure that are circulating into the body are also captured. So this tooth data can provide a fingerprint of the exposure. In our population, we use deciduous teeth, so baby teeth, and here is a description of what we have in our population. We have on the x-axis the weeks since birth, so we have weekly exposure of lead, and we have lead levels log transform on the y-axis, and 0 indicate the week of birth, negative value indicates the weeks before birth, and positive value on the x-axis indicate the weeks after birth, so the exposure after birth. Approximately we have 8 months of exposure here in weeks. We use the PROGRESS study, which is the Programming Research in Obesity, Growth, Environment, and Social Stress study, which is a birth cohort that initially aimed to identify the role of early life exposure on children's growth. And we enrolled 1,000 healthy pregnant women, and we follow them up with their children every 2 years. And it's still ongoing after 20 years. So we have the classical routine vaccination in this population because they follow the same schedule that we have in the United States. And in this case, we evaluate the association between lead exposure and the routine vaccination of measles, mumps, and rubella, and diphtheria, tetanus, and pertussis. And we are able to collect blood draw at 4 years of age and then assess IgG antibody levels of those vaccinations. As you can see in the table at the bottom, the majority of the children have antibody levels at or above the clinical correlation of protection. So those are healthy children. But as you can see, there is variability. On the top, we can see the log transform antibody levels. And because, of course, the majority of them are at above or above clinical correlation of protection, we use log transforming lac transform antibody levels throughout the analysis. So we use dynamic models, distributed not linear, distributed lag nonlinear models, and here is what we see. We have on the x-axis the time in weeks when the exposure was assessed. On the y-axis, we have the estimated association between lead exposure and the antibody levels in percentage, so change in percentage. So if we zoom in, we can see that around birth, a doubling increase in lead exposure is associated with decrease in antibody responses for diphtheria and tetanus vaccination of approximately 5% each. Despite lead exposure is sexually dysmorphic, we did not find any differences by sex. We have also, arsenic also caught our attention. Arsenic is mostly ingested through food, rice, and drinking water. And climate change is significantly increasing arsenic exposure in rice due to changes in soil chemistry that facilitate the arsenic absorption in rice plants. Here we have the early life exposure to arsenic in our progress children. I haven't said it before, but those children live in Mexico City, where the food consumption of rice is basically daily. So they are daily exposed to rice. And so this is why we're actually interested in testing also the arsenic exposure. And we saw basically similar association that we saw with lead. So around birth, a doubling increase in lead in arsenic exposure was associated with decreased antibody responses, in this case for measles and rubella. And as you can see, the window, the critical window of exposure in this case is larger than what we saw with lead exposure. And in this case, the association is, the average association here is approximately 7% decrease in antibody levels. But we are exposed to several chemicals at the time. So, and when we considering one chemical at the time, we are increasing the spurious findings. So we have to accommodate for the correlation among chemicals and identify the association of all these chemicals with the outcome. And this can be done using mixer methods. But there is only one single method that is able to capture critical window of exposures, multiple exposure, with a continuous outcome. This approach is the three distributed lag models, and it's a combination of mixture and tree-based methods that creates different partitions of the exposure over time. And it's also able to assess the marginal association for each exposure with the outcome while accounting for all other exposures. In this case, this method is able not only to accommodate for the correlation between chemicals, but also within chemicals, so between timing of the same chemical. We also consider other suspected immunotoxic chemicals, which are manganese, which is an essential metal, and cadmium, which is considered a toxic chemical. And when we use the specific tree-based distributed lag method, we see that late pregnancy is a critical window of exposure for cadmium, while accounting for all the others. And in that window, the cadmium exposure was associated with decreased antibody levels in anti-diphtheria antibody levels. So in summary, late pregnancy and weeks surrounding birth are critical windows of exposures that are associated with a decrease in antibody response to routine vaccination in school-age children living in Mexico. So those exposures can alter the balance and the responses of T helper cells that also are inducing a change in cytokines responsible for the production of antibodies. And this is what we are actually seeing. Now let's switch a little bit here and talk about white blood cells. Those are key players of the immune system. But we want to talk about the composition of white blood cells that is able to reflect the relative proportions of different types of cells within the same biological medium. So alterations of cell type composition might give information of the potential effects of immunotoxicants. And also investigating these cell type of alterations can be particularly informative during exposure windows like surrounding birth, when this regulation might act as an indicator of poor health, like birth outcomes or immunological outcomes later on. However, the distribution of cell types is not always available because it has to be measured in freshly drawn samples. And this has several technical limitations. The first one, of course, we require a large volume of fresh cells and also is very time consuming. So, the cell type distribution can be also estimated in bank samples using epigenetics. And when I speak about epigenetic in this talk, I'm considering the most studied epigenetic marks, which is DNA methylation levels, a specific genomic site. So just to give a brief and naive description of what is DNA methylation. Basically, there are chemicals that are attached to the DNA that can turn on and off genes, so they are able to regulate the gene expression without changing the DNA sequence. And DNA methylation levels at specific genetic sites, which are called CPG sites, offer a new way to estimate the proportions of cell types from bank samples. And prior studies have considered the association between the exposure and cell type proportions. So in this study, fine particulate matter, so PM2.5 exposure, was linked to all these leukocyte levels. And we found that the PM exposure level were negatively associated with the plasma, naive CD8, and natural killer cells, but they were also positively associated with naive CD4 and CD8 T cells. They also evaluated other exposure like black carbon, ozone, and temperature in the same manuscript. But in all these associations, the cell types were treated as independent, and so the exposures. So, but each sample represents a composite measure of cell types. So here we have a description of the cell proportions per sample, and we have 6 individuals, and we have blood, whole blood cell type proportions here, and we see that all the cell type proportions sum up to 100% or 1. And also the cell type proportions vary across individuals. So what we are expecting is that if one cell proportion is going down, other cell proportion are going up in terms of percentage. So so basically, this problem that I described, I call the mix and match cake issue. So basically, when we are creating a mix and match cake, this process involves combining multiple flavor cakes, and we have to select slices from the same set of cakes, and slices are proportion of a whole. But slices can be different across individuals based on preferences. So in both cases, the mix and match cake issue and the cell proportions, we have a sort of mixture summing up to a whole. So in this case, we consider the cell proportion as the outcome. And we want to test the association between environmental exposure and the specific outcome, which in statistics is called a compositional outcome. Both placenta and core blood molecular marks have been proposed as mechanisms underlying the effect of environmental exposures on birth outcomes, birth or childhood outcomes. But the effect of this environmental exposure on placenta or core blood cellular composition has been under-investigated. So we hypothesize that gestational exposure to fine particulate matter influences placenta cell type composition. So we are trying to see the association between one single exposure and the specific outcome, a compositional outcome, which is basically a multivariate outcome with a constraint of summing up to 1. In this case, we use 226 mother-child pairs from the PRISM study, which is a birth study, a multi-ethnic study as well, and in which we have trimester-specific fine particulate matter at the participant address. And we have also DNA methylation in placenta, and so we are able to estimate the cell type proportion in placenta. And of course, here in this plot, we can actually see on the x-axis the individuals, and on the y-axis we have the proportions of cells. As you can see, the blue indicates the most abundant one, which are the syncytial trophoblasts, and we order this plot by syncytial trophoblasts. We represent the association using the percent change of cell type per interquantile range increase in 5 particulate matter exposure. So actually, when we run this compositional regression, considering the 3 trimester of exposures, in the same model, we did not detect any statistical significant association between all the cell proportions and the exposure in the overall population. As you can see, the p-value are not statistically significant, greater than 0.05. But when we stratified the analysis by sex, we were able to identify that an interquantile increase of PM in the third trimester was associated with changes in placental composition. As you can see, the p-value is statistically significant, and this was driven by a decrease in nucleated red blood cell proportions that are non-mature cells that we are able to find in placenta. And in males, we see that an interquantile range increase in PM in the first trimester changes placental cell composition. And this was driven by a decrease in syncytial trophoblast and an increase of trophoblast. So we found that sex and time varying association between the fine particular matter exposure and placental cell type composition, suggesting compromised oxygen and nutrient exchange in placenta. And this is consistent with prior studies linking fine particular matter exposure and impaired placental vascularization and development and reduction of red blood cells in placental labyrinth zone. But in this study, we didn't consider any environmental mixture. So we decided to develop something with mixture as well. So in this case, we have a mixture of exposure and a mixture of outcome. In the progress study, we also have DNA methylation levels assessing cord blood. So we were able to assess the cellular composition in cord blood. And here we have the plot describing the cellular composition in cord blood. As you can see, the granulocytes, so the blue, are the most abundant one. And we want to assess the effect of the immunotoxic chemicals, which are arsenic, cadmium, manganese, and lead that were previously discussed in association with the cord blood cellular composition. Of course, we hypothesized that this gestational exposure can have an influence on cord blood cell types. We use an extension of weighted quantile sum regression. This mixed approach is able to estimate a weighted index summarizing the overall exposure, where the weights determine the contribution of each chemical to the mixture. And here we have a description in the plot A. And in the plot B, we see how this mixture identifies the association between the overall exposure that we see on the x-axis and the outcome that in this case we see on the y-axis of the plot B. So basically we extended this method. As I said before, we have one single outcome in this case, in this the traditional weighted quantile sum regression. But in our case, we have a compositional outcome, so a multivariate outcome with a constraint summing to a one or 100%. So when we consider the overall progress study, we did not find any association. And let me describe here this plot. In the previous analysis, we show the tables, but I thought plots can be very helpful to also understand the cell type, the proportion, the average cell type proportions, and how much is the shift changing 1 unit of the mixture. So in this case, we don't see much because we have black dots representing the average cell type proportion before any shift of the mixture. But then we have white dots that indicate the shift after a change of the mixture of exposure. As you can see, we have 7 outcomes. So for each cell type proportion, for each cell type, we have the effect of the mixture. But when we stratified the analysis by sex, we were able to see that we have a statistical significant association. So for a quartile increase in the mixture, we have a decrease in granulocyte for female, while an increase in granulocyte for female while a decrease in granulocyte for male. And also we have an increase in CD8 T cells and a decrease in nucleated red blood cells, a natural killer for female, due to a quartile increase in the mixture. And also we found that the mixture contribution, the contribution of each chemical to the mixture was different. And specifically in the male analysis, we found that cadmium was driven this association, while for female, the association was driven by arsenic. So, in summary, the gestational chemical exposures contribute to changes in cell type composition at birth. And these might partially explain the mechanistic connection between the exposures and the adverse birth outcomes or immunological outcomes later in life. Our findings were consistent with prior results, showing the maternal arsenic, lead, and cadmium exposure has been associated with lower level of T helper cells and induce systemic inflammation that also reflect changes in cell composition. So we also find sex divergent results, and this is potentially due to different circulating hormones and sex dimorphic effect of the exposures. Of course, as a future directions, we want to assess the persistency of those associations over time, but also evaluate whether the shifts in cell type composition mediated the relationships between the exposures and the aspect of the immune system at later stages, like antibody response to vaccination. And finally, we would like to determine whether those effects are consistent across ethnicities. I went really fast today. I want to thank all my collaborators that helped me to finalize these projects and also the fundings from the NIH. And I'm also happy to take any questions.
Well, thank you, Elena. This was a great talk. So we're open for questions. We can type the question in the Q&A box, or we can also unmute you. So, Elena, what do you think about like the protective effect of diet and nutrition, and how do we include those in mixture models?
That's a very great question. So we found that folic acid, for instance, has been protective. Actually, has shown beneficial effect when it's supplemented, and we have exposure to those chemicals. of course, I totally understand that manganese is an essential metal, and this can be also supplemented. Of course, we have to make sure that it's not over the threshold of beneficial threshold. But in this case, especially for WQS, we didn't consider any nonlinear association in this context because we had already several complications, including multiple outcomes and multiple exposures. So we didn't want to complicate this study too much, but we have also evaluated the effect of folic acid, stratifying the by those those women that supplement with folic acid their diet. And in our case, we didn't find much. This can be potentially due to the limited sample size that we have, and of course has to be validated in other population. Also, it can be due that the association is not directly linear, and so this is something that we didn't account for, but it will be lovely to test it.
Yeah. Do you have any plans for the replication cohorts? Like follow-up study to replicate these findings in a different cohorts?
We are thinking of it, but it's very challenging to find studies with all this type of data, especially with teeth. It's very challenging to have both antibody response to vaccination and teeth data, environmental data. And so it's a very unique cohort. It's a very unique population. And we have also follow-up every 2 years. So this is why it's so precious to us.
Yeah. Any questions from audience? Oh, there's one.
Yeah. I'm unmuting them now.
In the meanwhile,
Yeah, Nancy, you have a question.
Let's go back to Nancy in a minute, so I'll answer the other question that we have in the chat.
Okay.
Are you planning a multi-omics approach? This is a great question. I would love to have a multi-omics in this population. We have a few omics, and we are considering to evaluate multi-omics as mediators for the association between exposure and outcomes. So yeah, we are considering that for future analysis. Nancy?
Nancy, do you mind typing the question in the Q&A box? If it's quick, yeah.
So for the layer that we are considering definitely DNA methylation because we have it, and also metabolomics as well for now. And Nancy, we are waiting for your question in the Q&A.
What are the metal concentration ranges in progress? Helena, do you know? That's Nancy's question. Exposure range, concentration. Yeah.
Sorry, I didn't say that. Sometimes when I give things for granted, so like this. So the exposure level, especially for lead levels, are higher because the progress study is uniquely exposed to lead because it's it's a Mexican population, and they are exposed to lead glazed pottery. So on top of the exposure that we currently have in the United States, they have this unique exposure. And so it's a little bit higher than the United States, but comparable with some of the population with low social economic status in the United States. And for arsenic levels, we have a similar levels in the same ethnicity that we have. So in the Mexican and Hispanic immigrants, we have the same exposure level in Progress and in the United States. So for the other metals, I haven't checked. I don't recall, honestly, and I'll try to fill this gap in the manuscript that we are finalizing. So.
Thank you. Any other questions? All right. Okay, so thank you, Elena, for this great talk and very exciting. Oh no, we have a question from. We still have time, so we can take one more question.
Yeah, sure.
I saw a raised hand.
Can you hear
Go
me
ahead.
fine?
Yes, yes.
Okay.
Yes.
By any chance, like, are you also considering microbiome data from the patients? Because there is a lot of like commotion around it, and with exposure, it would be a great option. Or already maybe it is available and I missed that part.
I don't usually work with microbiome data. I know that compositional regression is very popular within the microbiome world. I don't work with that, but I know that others have experience with this type of data. I don't know. This is a great question. Definitely something that we can investigate and more on this. Yeah, but this is a great question. If you have a great population, you can potentially do the same.
Thank you so much. It was a great talk.
All right. So thank you again, Elena. Can we bring the last slide? Because this is a final webinar for the HERE Exposure webinar series. And so I want to thank you all for attending. All the webinars are recorded and available on NIH's YouTube channel or the HERE program site, hereprogram.org. And they are available prominently there. There are about 21 of total, covering different topics for the exposomics. So with that, we are concluding the HERE webinar series. Thank you all for attending.